Yeah, go ahead.
All right, everyone. So today's topic is connective and supportive tissues. So basically, a connective and supporting tissue consists of a small number of cells which are present in large amount of extracellular fluid. And this extracellular fluid consists of fibers, ground substance, and tissue fluid. And it is also made up of protein fibers. Uh, like given here, it says collagen type 1, 3, and elastic fibers, and it also consists of amorphous substance. Now, the three types of ground substances are present depending upon their consistency, so that you can classify the connective tissue further into three types. So, if the ground substance is going to be a bit fluid, it is going to be classified as connective tissue proper. And if it is a bit more firm, then it's cartilage. And if it's completely calcified, then it's going to be bone or cement or dentin. So all type of connective and supportive tissue is always going to have these three things, which include cells, fibers, and ground substance. So these cells, they are specific for each type of connective tissue. So what this basically means is that if you're looking at adipose tissue, for example, then only adipocytes are going to be found in this particular type of tissue. So these cells, what they do is they help in production of the matrix and also the fibers and the ground substance. So fibers are as well specific for each type of connective tissue. Uh, this will be explained to you later on in the PPT. And ground substance has a similar composition in all type of connective tissues and cartilages. It does not really fluctuate much, and but the only difference is that it is mineralized in hard tissues. Where do these cells come from? So there are three origins to all of these cells. There's mesenchymal stem cells, hematopoietic stem cells, and ectomesenchyme of the neural crest. The these all the examples given here are really important in the sense of your micros because they can ask you where these cells originate from, and you must know. The important thing over here is all but cell types originate from your hematopoietic stem cells, and there's also something called as wandering cells. These are the examples. So wandering cells basically mean that these cells are not always present in the connective tissue. Um, Depending upon if you have like an infection or immune response, according to that also they can like come and go, so they're not always going to be found in your connective tissue. And over here, ectomesenchyme of neural crest cells give rise only to the odontoblast, which is an important thing because this is the only thing it gives rise to, so it is kind of like an exception. So this could also be asked in one of your tests. Now, let's talk about our connective and supportive tissues. They are further classified into four things. Embryonic mesenchyme is basically found during your embryonic growth, and it gives rise to connective and supporting tissue when you are still like in your embryological developmental period. Now, when we talk about our connective tissue proper, it is further divided into loose connective tissue and dense connective tissue. Now, dense connective tissue is again divided into regular and irregular. So when you look at the diagrams given here on the right side, the leftmost over here is going to be your regular dense connective tissue, because you can see that it is, compared to the other two, it is like more organized in a way, and you can see that all the fibers are only going in one direction. So that is when you know that it is a regular sort of dense connective tissue. Now, I have a question for you guys. Out of the other two pictures over here, which one is going to be irregular dense connective tissue and which one is loose connective tissue? Any ideas?
Regular is the one on the left.
The one on the left is which one? Sorry.
Regular?
No, regular is the leftmost, which I already spoke about, but I'm asking about the
Oh,
picture in the middle and the left one. Sorry, the right one.
The one
So,
in the middle is the irregular dense, and the one on the right is the loose.
yes, exactly. That's very good.
I
So,
said irregular.
oh, sorry, my bad. I completely misheard it. So, the basic way you can identify this in a generic sense is right below your epidermis. You're always going to have a loose connective tissue because it is kind of like an interstitial connective tissue. So that's the first layer which comes, and then it slowly changes into dense connective tissue. But this kind of a classification is not like it can be kind of difficult for you to identify it. So there is another way you can look at it, is that loose connective tissue over here, as you can see, it has got a lot, it's more nucleated compared to the dense one. Like here you can see there's a lot more these purple spots which are the nuclei. Compared to the dense one, this has more, so that's how you can also identify it. And let's move on to connective tissue with special properties. So there's going to be four types of them. There's the adipose, elastic, reticular, and mucus. Now your adipose tissue is also further divided into white and brown. So any ideas? This is a very basic question, but could you figure out which one is white and which one is brown looking at these pictures? Anyone? All right, so the one on your left is your white adipose tissue because like you can see here, this you can only see white with like a very few demarcations between the cells. And on your right side, it's more brown because there's like less droplets of lipids in them. So that is why this is going to be your brown adipose tissue. Now, moving on to the elastic. Um, the major thing to note here is that it is going to be found in the elastic membranes of large arteries, and this will be taught to you in detail when you come to your cardiovascular system. But for now, for your micros, they can give you a picture of the connective tissue, and you might be able, like, you will be expected to answer what it is going to be like. So for here, when you can see this picture over here, this is going to be your elastic connective tissue, and the way you can identify it is you can see these purple lines here, and they are sometimes like a bit wavy. So, and this is going to be very prominent in any slide you see or any photograph which is given to you as well. Whenever you see these kind of lines, dark lines, which are like a bit wavy, that means you're looking at elastic connective tissue. And when you talk about a reticular connective tissue, it is a supporting sort of a tissue which is found in your parenchymatous organs. And this will be again explained to you further on when you do your lymphatic system. But again, for now, you must be able to recognize it. So when you look at this picture over here, that is a picture of a reticular connective tissue. And the way I remembered it is that it looks like a cherry blossom, kind of. So that's how I always knew that I'm looking at a reticular connective tissue. The way it looks in the photographs and the way it looks actually in a slide is a bit different. So for now, you don't have to worry about that. All you need to know is how it looks like in a photograph, and you should be able to identify it based on that. Let's about supporting connective tissues. They're further divided into three types. There's three types of cartilages: hyaline, elastic, and fibrocartilage. And there's bone, and there's going to be dentin and cementum as well. Now, the connective tissue proper have two components, cells as well as extracellular matrix. So your cells are further divided into fixed cells and wandering cells. Like I said earlier, fixed cells are all of these cells given over here, which are always going to be present when you're looking at a specific type of connective tissue, depending upon which tissue you're looking at, of course. Now, if I'm looking at only adipose tissue, you can see, you will only notice adipocytes, which is always going to be present. And when it comes to wandering cells, like I said earlier, they are not always present. They can come and go depending upon what kind of a situation your body or that particular organ is going through. So this one, all of these examples are pretty important because they can give it to you in a micro test, like which of the following is a wandering cells, and you must be able to remember all of these. Now let's talk about the mesenchyme. uh, so it is like a sort of an embryonic connective tissue which arises from the mesoderm when you're still in your embryonic periods of life. And these cells, as you can see here, are kind of like star-shaped cells which form a 3D network. And as you can see here, that they have got a really prominent nucleoli present in the nucleus, and the cytoplasm is pretty sparse. It's not that well present over here, and you can see that it extends like a lot of processes which kind of form a 3D network. These cells are highly mitotic, uh, and their intercellular spaces are filled with like a gel-like substance which contains scattered reticular fibers. And now I'm going to let Katerina continue with her slides.
Thank you. Does anyone have any sort of questions for the part that we just covered? No? Okay, then we'll move on. Okay. So now we are going to talk about cells of connective tissue proper. So as Ria explained in her previous area, I hope you guys understand that we're talking about three different types of connective tissue. We have connective tissue proper, connective tissue with special properties, and then we have supporting tissue. So right now this section is going to focus on the cells of connective tissue. And I also hope that you guys understand the important part, which is that connective tissue in itself is always going to be composed of three things. It's always going to be composed of the cells, the fibers, and the ground substance. This is very important for you guys to keep in mind because they will ask you these things in your slide tests and in your final. So now we're going back to cells of connective tissue proper. The most common, and one could argue one of the most important cells, because you guys will see them all the time, are these cells called fibroblasts or fibrocytes, which you guys will also see. So these fibrocytes are the most common cells of connective tissue proper. Fibroblast is an active cell, and it synthesizes the extracellular matrix, which, as you guys know, is a very important component of connective tissue. So again, fibroblasts are active and they synthesize the extracellular matrix. And the extracellular matrix is made up of protein fibers, collagens type 1 and 3, and elastic fibers and amorphous substance, which you guys will understand later on what these things are. But for now, just remember that fibroblast is active. Fibrocyte is a mature resting cell, so it does not have any synthetic activity. However, after it becomes stimulated, the fibrocyte can actually return to its fibroblast state and start synthesizing extracellular matrix. That's why here I have these arrows going both ways, because fibroblasts can turn into fibrocytes and vice versa. Both cells are spindle-shaped, and they have few cellular processes and an ovoid nucleus. Now, the fibrocyte is smaller than the fibroblast. And my question to you guys is why? Do you guys know why the fibrocyte would be smaller than the fibroblast? Mind you, the answer has already kind of been mentioned, but does anyone know why?
Because it's resting.
Exactly. Fibrocyte is smaller because it is rusting. The fibroblast is an active cell, so you have a lot of different cellular processes occurring, which is going to make it larger. So here is an example. I think it's kind of obvious. I would ask you guys to guess which one is the fibroblast and the fibrocyte. In this picture, like which one is this?
Fibroblast.
Fibroblast. As you guys can see, you have all of these cellular processes ongoing while the fibrocyte is mature and resting. Now, here on the right is what you guys would be able to actually see in your tissues. It is very difficult to sometimes kind of, even with the microscope, to properly see a fibroblast versus a fibrocyte, but in the scenario that it is possible, this is what you guys would probably see. So here you can see, you know, kind of a difference. I'm guessing this would be a fibrocyte and something larger would be a fibroblast. Even if you're not able to properly notice it on the slide, if you can explain to your professor, like theoretically, well, the fibroblast is going to look like this because of that, and the fibrocyte is going to look smaller because of this. If you can explain it theoretically, the professor will let it go. So we also have reticular cells. So reticular cells are cells of, as the name says, reticular connective tissue, and this is found in the spleen, lymph nodes, red bone marrow. And reticular cells, these cells, they create a stroma and they offer structural support. Reticular cells are technically a type of fibroblasts, but they're fibroblasts that are specialized to produce type 3 collagen, which are reticular fibers. And they also produce amorphous substance. So remember, reticular cells are a type of fibroblasts that are specialized to produce type 3 collagen. Type 3 collagen makes reticular fibers. And they are stellate-shaped with long processes. So the next are myofibroblasts. Myofibroblasts are usually found during wound healing in connective tissue. So if someone were to cut themselves in their hand and it would be healing and you would analyze that sort of tissue under a microscope, you guys would see a lot of myofibroblasts. Myofibroblasts are similar to fibroblasts, but they have a lot of myofilaments. Now, you guys have not done muscle yet, so you might not know what that is, but just try to remember that myofibroblasts has myofilaments such as actin and myosin. And the function of myofibroblasts is that they're responsible for wound closure And they do this by contracting or by contraction. So just try your best to remember that, because this is something that I know some professors really like to ask: what is the function of myofibroblasts? And always try to remember the name. A lot of times the answers are in the names. You have reticular cells, you know that they're going to be responsible for reticular fibers. There's the word myofibroblasts. These cells, the word fibroblast is there, but the word myo is there. You're going to connect myo with myofilaments, and you're going to know that it's going to be responsible for contraction and wound healing. You have adipocytes, which Ria already mentioned before. So these are found in adipose connective tissue, and you can usually find them either in isolated adipocytes, where you see one adipocyte, or you see them in small groups, which is actually much more common. And Ria mentioned it, but we can talk a little bit more in detail. There are two types of adipocytes. You have unilocular white and multilocular brown adipocytes. Unilocular, it's one, so it's a single large drop of fat, and this will have a flattened nucleus and a small amount of cytoplasm. The function of the white adipocytes: nutrient, heat, structure. This is something that you will be asked in your slide test. So remember, the function of white adipocytes: nutrient reservoir, heat production, and they offer structure. And you guys have already guessed it, but just more reinforcement. Here, we're looking at white adipocytes. As you guys see, it's one single large drop, and it will have a flattened nucleus and maybe a small amount of cytoplasm that you might see, but might not see, but just know theoretically. And again, nutrient, heat, structure. Multilocular, or the brown adipocytes, the cytoplasm will have a high number of lipid droplets of various sizes. So here you go. And inside, you guys can see all of these lipid droplets. Here we have one cytoplasm with one, while here you guys can count, I don't know, I'd say like maybe 10-ish, 15, I don't know, maybe more. Many lipid droplets in one cell of various sizes. You're gonna have abundant mitochondria and lysosomes, which gives it its brown color. And know that this in humans is only present in fetuses. And it's important for protecting the newborn against the cold. So you guys are not going to find these in adults or children's or like nothing. You'll only find these in very, very young newborn children. Now we have monocytes, macrophages. So in connective tissue, try your best to not get confused, because this can be a little bit confusing. Because all of these are the same, but depending on where they're located, we call them different things. So in the blood, we have monocytes. When they're in the blood, they're called monocytes. There are blood elements, and these monocytes are precursors of macrophages, meaning they turn into macrophages. Now, when do they do that? They do that when they have migrated from the blood into the connective tissue. So monocytes are blood elements, and once they have migrated from the connective tissue into the blood, they're now termed as macrophages. Oh, sorry, once they've migrated from the blood into the connective tissue, they're now termed as macrophages. So these macrophages in the connective tissue, we also call them histiocytes. So monocytes, macrophages, histiocytes, same thing, but just different names depending on where they're located. Connective tissue, macrophages, histiocytes. Monocytes are once they're in the blood. And these macrophages, as we see them in the connective tissue, they have an immunological function. So they ingest any foreign particle, and they function as antigen-presenting cells. I know you guys don't know what this is yet, because you haven't done the immune system yet, but just know that macrophages have an immunological function. They ingest like foreign bacteria, like anything that we see that it shouldn't be there, or anything that shouldn't be there in our body, they ingest it, and they present it to lymphocytes to be destroyed. So they also secrete substances that participate in defensive and reparative functions. They're also members of the mononuclear phagocytic system. Again, I know you guys don't know what that is yet, but you will know later on. We have mast cells. Mast cells, just know that they are in the dermis of your skin, and they're also in your digestive and respiratory tracts, and they're involved in some immune response. You also have plasma cells, and plasma cells, They are seen in areas of chronic inflammation, and they also have immunological functions. Now we're going to talk about collagen fibers. So here we have the main collagen fibers that you guys are going to be asked about and that you guys are going to see constantly or most of the time. That's type 1, type 3, and the elastic fibers. So type 1 collagen, it's produced by fibroblasts, osteoblasts, cementoblasts. I know you guys don't know what the last two are yet, but just kind of go with the rhythm. Fibroblasts, osteoblasts, cementoblasts, they will all secrete type 1 collagen. And just know that first you secrete procollagen. Procollagen is soluble, right? And soluble is not going to work because it's not going to able to maintain the tissue structure that we want it to maintain. We can't have something soluble. We want it to be insoluble. So we went from procollagen to tropocollagen, and then there's some polymerization to kind of give it the hardened structure that we want. Later, once that's made, it's going to start aggregating into the collagen fibers and form bundles of collagen fibers. And the bundles are what we actually see when we're looking at the slide. This last part is very important because this will be, if I'm not mistaken, part of your first credit test, because you guys are going to do histological techniques. I know you guys don't know much about how the tissue is actually prepared, but in order for us to see the things that we see, the connective tissue, the epithelium, the glands, they all need to be stained with certain stains. So for us to be able to see collagen really, really well, we need to stain it, or we can stain it, or it's usually stained with hematoxin and eosin, Masson's green, or blue trichomes. If you guys know this for your first slide test, professors are absolutely gonna love it because they all love histological techniques. Now, type 3 collagen is also produced by fibroblasts. However, if you guys remember, reticular cells, right? So it can also be produced by reticular cells and smooth muscle cells. Same thing, procollagen, tropocollagen, and then there's polymerization. It will be aggregated. However, not in bundles, but in a network. Because remember, the reticular forms a network. So reticular fibers are going to form a network, not a bundle. And you guys will understand later on when you do the immune system, when you look at like the spleen and lymph, why this network is important. It's important because it's going to trap like debris and bacteria so they can be eliminated. So remember, reticular network. And this can be visualized by the Gomori impregnation technique. So this is the histological technique. And then we have the elastic fibers. So the elastic fibers are produced by fibroblasts, reticular cells, chondroblasts. And here it's a little bit different. You have glycoprotein in the microfibrils, or globular protein proelastin, that will then polymerize, and you're going to have this amorphous rubber-like elastin. This may form a network and can be stained with orosine or resorcine fusion. And then you have type 2 and type 4 collagen. These ones are not, I won't say are not as important. You 100% can and probably will be asked about it. Type 2 collagen, it will form only very thin fibrils, very, very thin fibrils. They do not form fibers, only fibrils. And it's a very submicroscopic structure. They display cross-banding, and it's present in the matrix of hyaline or elastic cartilage. And type 4 collagen do not form fibrils or fibers. It's just present in the basal lamina of epithelium. And it's produced by epithelial cells, muscle cells, and Schwann cells. Okay, and next, Varun is going to talk to you guys about the types of connective tissue. And sorry, guys, any questions regarding what we just went through? Okay.
All right, guys. So let's talk about the types of connective tissue. So just a revision question. Can anyone tell me all the types of connective tissue?
Loose and dense connective tissue.
Yeah. Anyone else?
Regular, irregular.
Yeah, that's the classification of the dense connective tissue. All right. So first we'll
I think you are muted.
All right. So first, we'll begin with the loose connective tissue. the areolar connective tissue. So first, I want to emphasize on the word areolar because sometimes in your microtest, they don't mention loose connective tissue. They can just say areolar connective tissue. So just make sure that it's the same thing. So first of all, where is it found? It's found in the skin. It is found in the mucosal layers such as tunica subserosa in the gut, which would be in the stomach, as well as the intestines, as well as the dermis. And now, what's the function of it? It serves as the padding and the packing material, which provides flexibility and support. So, what does this mean? So, basically, the loose connective tissue, it acts like a cushion. So, it will be like a cushion around your organs, and it will protect it from getting crushed. Now, when we talk about the components, we have the cells which are divided into two categories. We have the fixed cells and then we have the wandering cells. The fixed cells include the fibroblasts and the fibrocytes. As Ria mentioned before, fibrocytes are involved in the production of fibers and the ground substance. Now, when we're talking about the wandering cells, these include the macrophages, plasma cells, mast cells, lymphocytes, and granulocytes. And as Ria said before, they can ask in your micro that what of the cells from the given option are wandering cells. So here you can use this mnemonic to memorize them. Many people make life great. So obviously M for macrophages, P for plasma cells, M for mast cells, L for lymphocytes, G for granulocytes. Now, what is the function of wandering cells? Basically, they act as, soldiers or just like protective guards of the loose connective tissue. So they make sure that they are ready to fight off any bacteria or germs that enter the loose connective tissue. Now, uh, yeah, wait, yeah. So what type of fibers do you guys think are present in the extracellular matrix of the loose connective tissue? By the type of fibers, I mean collagen fibers, elastic fiber, reticular fibers. What all of them are present in there?
All of them?
Yeah, exactly. So the extracellular matrix, it contains different fibers, elastic fibers, collagen fibers, as well as reticular fibers. So all these different fibers, they have different roles. Elastic fibers will help in elasticity, collagen fibers will provide strength, and reticular fibers, they would form like, a spider-like network. Now, then it has the ground substance. The ground substance is abundant and facilitates exchange of substances. As I had mentioned previously in the previous seminar, the ground substance is like, a jelly-like substance which fills spaces between the cells, and it allows the cells to remain in their place as well as provides them with nutrients which are needed for their growth or sustain their life. Now, just to summarize, the characteristics of the loose connective tissue would be it has variety of cells, including both fixed and wandering, few fibers compared to the other connective tissues, and abundant ground substance providing loose and flexible support. So, just to summarize it, the loose connective tissue, it is like a flexible cushion that supports and protects other parts of your body while letting other things move freely. So you can see in the diagram, this whole empty space is the ground substance. This whole cell is the fibroblast, and then these are the elastic fibers, and then these are the collagen fibers. All right. Now let's talk about the irregular dense connective tissue and the regular dense connective tissue. So Ria mentioned this before, but I still want to ask, what do you guys think is the difference between the irregular and the regular dense connective tissue? All right, then let's start. So the location of the irregular dense connective tissue would be that they're found in the reticular layer of dermis, as they're found in capsules of organs such as in perichondrium and periosteum. So what is perichondrium? Perichondrium is nothing but a layer of fibrous dense connective tissue over the cartilage, while periosteum is the layer of fibrous dense connective tissue over bones. Steum means bone, chondrium means cartilage. Now, what's the main function of this is that it provides structural support and resists stress from multiple directions. All right. Now the cells are again the same as the loose connective tissue. That would be the fibroblast and the fibrocytes, which again have the same function, provide and maintain collagen fibers and extracellular matrix. Now we come on to the extracellular matrix, which contains fibers and ground substance. So the fibers which are present here are the collagen type 1 in coarse bundles, and they're arranged without, a uniform arrangement. And the ground substance present here is less abundant compared to the loose connective tissue. And again, it has the same role, jelly-like structures, fills the spaces between the cells and providing support. So now, what is the use of this, the arrangement of the collagen type 1 bundles in a non-uniform arrangement? Basically, what this means is that this type of irregular connective tissue, wherever it is present, yeah. So these collagen type 1 fibers, they're arranged in, a crisscross arrangement, all right? So if you pull this irregular dense connective tissue from any direction, yeah, it will resist that. So it is flexible yet strong type of connective tissue. Now, basically, this is the same thing, characteristics dominated by bundles of collagen fibers with no specific orientation, allowing it to withstand stress from different angles. So no matter what direction you pull this from, it will still withstand that stress. Now, let's talk about the regular dense connective tissue. So when we talk about the location, it is found in tendons as well as ligaments. So tendons are basically connecting muscles to the bones and ligaments are connecting bones to the bones. So the main function is providing strength and resistance to pulling forces in one direction due to the parallel arrangement of fibers. Now, as I said before, irregular dense connective tissue resistance from all direction because of the crisscross arrangement of the coarse bundle of the type 1 collagen fibers. However, in this sense, there is, uniform arrangement or parallel arrangement of the collagen fibers. It will only resist pulling forces from one direction. Now, we talk about the cells, fibrocytes, which basically again produce collagen fibers and ground substance. And the extracellular matrix, primary collagen type 1 fibers organized in parallel bundles, which provide it with strength and tensile properties. Ground substance, again, minimal, providing just enough matrix to hold the cells together and allow for a bit of flexibility. So there isn't much space between these fibers, which is why, this type of dense connective tissue feels firm and less flexible. This is why it is really good for keeping bones and muscle stable in their position and also allowing for movement. And then we'll talk about this characteristics would be the orientation of the collagen fibers is uniform, as I mentioned, and compared to the connective tissue, it has less elasticity but high tensile strength. Now we're talking about the elastic connective tissue. So the main areas where the elastic connective tissues are found would be the elastic membranes of the walls of arteries and veins and yellow ligaments of the vertebral columns. So the main function of this would be it provides flexibilities and allow tissues to stretch and recoil. So basically the elastic connective tissue is like a rubber band. So when you apply force to the rubber band, it will expand and it will change its shape. However, when you remove the force, it will return back to its normal shape. So that's basically the main function of the elastic connective tissue. The cells present are the fibroblasts and the fibrocytes, and it produces and maintains elastic and collagen fibers. Now, the extracellular matrix, it contains fibers which are dominated by the elastic fibers, and it contains the collagen fibers as well. So the main role of the elastic fibers would be providing the elasticity, allowing them to change their shape, while the collagen fibers provides them with additional strength to make sure nothing breaks apart. Ground substance, similar to regular and dense connective tissue, it is present, but it is not abundant. It provides support and is present between the fibers. So yeah, the main characteristic of these elastic fibers would be that it allows tissues to expand when needed. For example, blood vessels expanding with the pulse and return to their original space. So here you can see all these elastic fibers here, and this picture shows the elastic artery, which you'll see in the further lectures. And then now the last one would be the reticular connective tissue. So reticular connective tissue, it is a special type of loose connective tissue that forms a supportive network. It provides framework for lymphatic organs such as lymph nodes, spleen, and bone marrow cells. Now, it is made up of reticular cells, which are star-shaped cells that form a network-like structure. It sends out arms, which form spiderweb-like structures. So yeah, that. And then we have the extracellular matrix, which contains fibers, which consist of reticular fibers, which are also called type 3 collagen fibers. So you guys have to remember that reticular connective tissue, type 3 collagen fibers, irregular and irregular dense connective tissue, type 1 collagen fibers. This is really important. It will be definitely in your micros. One of the question would be which type of collagen fiber is there in reticular connective tissue, which is in irregular or regular connective tissue. Ground substance, again, minimal, acting or as a medium to support cells, provide them with nutrients and allowing the cells to remain in their place. So the main characteristics of the reticular fibers is that it creates a scaffold that supports and anchors cells like white blood cells and bone marrow cells. And it is also found in organs where you need to, filter fluids as well as help in immune function. So that was my part. Does anyone have any questions?
Okay. So next time, last thing that we're going to talk about is cartilage. It's not gonna be long, it's gonna be fast, but I just wanted to quickly go back to this because I feel like you guys might still be a bit confused on the classification. So connective tissue proper, guys. Loose connective tissue, dense connective tissue. Dense connective tissue, you have regular and irregular. Connective tissue with special properties, word being special, found in specific locations, adipose, has adipocytes. You're going to have elastic connective tissue that is going to be found in structures that are going to need to expand and then return to its original shape. You're going to have reticular, just like Varun just mentioned, support like white blood cells. And then you're going to have supporting connective tissue, cartilage, bone, dentin cementum, which is the one that we're going to talk about now. Like, this is the one that you guys are going to be seeing quite a lot, especially in the beginning. But please keep in mind that connective tissue with special properties is there, adipocytes, elastic reticular, and supporting connective tissue. So please try your best to not get confused with these things. So cartilage is supporting connective tissue because I just showed it to you guys. And let's talk about what cartilage is. So first, cartilage consists of the extracellular matrix, as we talked about, and it's going to have a fibrous component, a ground substance, and then the cells. So the extracellular matrix is going to be avascular, and it's going to be highly specialized. And under the extracellular matrix, you're going to have fibrous and the ground substance. The fibrous, you're going to have type 3 collagen fibrils, elastic fibers, and type 1 collagen fibers. The ground substance, you're going to have proteoglycans and glycoproteins, and the glycosaminoglycans are going to bind to type 2 collagen and form a cross-linked matrix to provide consistency. The cells, you're going to have stem cells, which are going to turn into progenitor cells, chondroblasts, and chondrocytes. Now, I know that like there are a lot of words here that just seem very repetitive and like it's not making sense. This is like a very technical information about cartilage. Just honestly try your best to memorize it and understanding it. There's really no other way for us to communicate this information to you guys. Just remember that extracellular matrix has the fibrous components, the ground substance. The fibrous component has fibers. So it's going to have elastic fibers, type 1 collagen fibers, and type 3 collagen fibrils. And the ground substance are going to have, it's going to have proteins. Proteoglycans, glycoproteins, etc., which are going to help give a consistency. For cells, you're going to have chondroblasts and chondrocytes. Remember fibroblasts, fibrocytes? Now we have chondroblasts and chondrocytes. And we can distinguish depending on the arrangement of the cells. So the chondroblasts, chondrocytes, and how much fiber there is, and how much ground substance there is. And depending on all of this arrangement, we can differentiate between 3 types of cartilage. We have hyaline, elastic, and fibrous cartilage. So a little bit about the origin of cartilage. Just know that it originates from mesenchyme. You guys will understand exactly what this is better when you guys do embryo, but for now, just know that cartilage comes from mesenchyme. And I put this here because these pictures might pop up in your slide test, in your micro tests and stuff. But just know that picture A, here we have the primitive cells, and they're aggregating to form a blastema. In picture B, the cells in the blastema are starting to divide, or multiplicate. And then in C, the cells will start to secrete this cartilage matrix. So here is the matrix. The cells are secreting this matrix. And once the cells start to secrete it, they're now known as chondroblasts. Chondroblasts secrete cartilage matrix, always. In D, we're looking at the mitotic division of the chondroblasts, and they're going to form isogenous groups. Now, one thing that's interesting for you guys to know is that, so we have chondroblasts, right? We have these cells. The cells are secreting cartilage matrix. And once they secrete this matrix, they actually kind of become embedded on it. It's like they become stuck on the things that they secreted. And once they become stuck to it, they differentiate into chondrocytes. So if you guys get asked, how do chondroblasts turn into chondrocytes? Is, well, the chondroblasts are going to secrete the matrix, they're going to get stuck in the matrix, and then they're going to differentiate into chondrocytes. Okay, so hyaline cartilage. This is how hyaline cartilage looks like. One of the secrets that the professors like to mention for you to easily recognize it is that it has a glass-like look to it. It looks like glass, like some sort of transparency. So hyaline cartilage is located in these areas, as you guys can read: nasal ala, laryngeal cartilage, trachea, bronchi. The cells, you have chondrocytes that are arranged in isogenous groups, which you guys saw, like what this means, isogenous groups. You have thin collagen type 2 fibrils. The ground substance, you see there's a lot of ground substance that's occupying like large fields between the isogenous groups. And you have the perichondrium, which is going to help provide like vascularization, innervation, and allow it to grow. So hyaline cartilage, it lacks the nerves, blood, and lymphatic vessels for that reason. Therefore, the regeneration potential is very limited. That's why like you need the perichondrium, which is going to help provide like nutrients and innervation and allow it to grow. You have elastic cartilage. Elastic cartilage is found in such areas. And guys, like a lot of histology is really just memorizing how things look like. Take a good look at the hyaline cartilage, okay? You see that it's always like, it looks almost like a glass, like transparent look versus the elastic cartilage. In the elastic cartilage, you guys can actually see all of the elastic fibers. So elastic cartilage is found in epiglottis, larynx, auricle, like things that actually require elasticity, and it's very resilient. It has a lot more cells than hyaline cartilage. The cells are also going to be chondrocytes. However, here they're not going to be in isogenous groups. They're either going to be single or they're going to be paired. And the fibers are going to be elastic and collagen type 2 fibers. Or fibrils, sorry, because collagen type 2 has fibrils, not fibers. And ground substance is lower than in hyaline cartilage because look at all the cells and all of the elastic fibers. Lastly, we have fibrocartilage. Fibrocartilage, you guys will not see it as much. I believe you might see it maybe in 1 or 2 slides, but it's found, or maybe none, but it's found in the intervertebral discs, pubic symphysis, etc. And the cells, you have up to 4 chondrocytes that are arranged in like elongated isogenous groups. Fibers, it's going to be collagen type 1. There's only going to be a very small amount of ground substance, and the thing that sets apart fibrocartilage from the other 2 types of cartilage is that it has no perichondrium. And now, do you guys have any questions from what we just covered? If not, we have a mock micro that you guys can now do, and I will stop sharing and stop recording now.